Okay, welcome to the next session. And so we have some great speakers for this session. The first is Professor Paolo Sassone-Corsi, who will be speaking on the epigenetics and metabolism, the clock connection. So Paolo is a professor and chair of pharmacology at UC Irvine in California. Prior to that, he was director of research at Strasbourg in France, and he's really made several contributions to our understanding of cellular signal transduction and mechanisms of control of gene regulation, including chromatin and histone regulation. And he's really a leader in the genetic control of circadian rhythm, including key regulatory events in histone regulation. So we'll welcome him, and you have the podium.
Thank you all. It's a pleasure to be here. I'm going to tell you a few things about the studies we've been conducting in our Center for Epigenetics and Metabolism. Occupies the top floor of this building at UCI. This is my group, and what I'm going to tell you today mostly is work done by, I guess, two ladies. This one right here, Christina Kelmahan, and this one right here, Summer Mastering. And we've been lucky enough to have the support by this fellow right here, sabbatical professor from Munich. His name is Axel Niemoff. All right, so the key question that we'd like to address in our center is how nutrition impacts on epigenetics. And I'll tell you very briefly about what is epigenetics in the introduction of my talk. The question about nutrition is critical, especially this time in most countries where epidemics of obesity and diabetes becomes dramatic. This is true specifically here in Saudi Arabia, as you can see from these slides. Saudi Arabia is one of the countries where obesity is at the maximum and is increasing with dramatic proportions. In fact, the rate of obesity increase in Saudi Arabia is higher than any other country in the world at this point. And this rate of diabetes, as you can see also, Saudi Arabia is in a bad situation. So I think the questions that we address in our center are highly relevant to this country. One really sad record of Saudi Arabia is the report of the very first, oops, okay, I didn't do anything. Maybe I shouldn't talk about that stuff. That's why it isn't here. Okay. Is the record of having a very first 2 years old obese child who went under gastric bypass surgery to decrease the rate of obesity and diabetes. Now, what is that determines all this and how this relates to epigenetics and specifically to the disruption of the circadian clock? That's what I'm going to tell you today. Now, the concept about epigenetics comes from this fellow. This is was an English developmental biologist, Corey Waddington. In the 50s, he came up with, okay, I don't know what to do, but I'm not going to touch anything. All right. He came up with the concept that equal cells with the same genome will become different only because they will be shaped by what he called an epigenetic landscape. And this is the basis of all stem cell biology these days. How is it possible that two equal cells will become either neuron or adipocyte? The idea is that the landscape will be different valleys and troughs in a specific landscape where the cell, here represented by marbles, will roll down, and the landscape will shape their genome so that they will function in different ways, and thereby determining the fate of those cells. How is that possible, and why is it so important that genome? I'd like to show this slide. This fellow is Kobe Bryant, and he trains at UCI, so I kind of meet him all the time in the coffee shop. And one day I realized that he's as tall precisely as much as DNA we have in one cell. So in one cell we have 6.5 feet of DNA, 2 meters, and that's how much Kobe is tall. That means that in his retina, he requires to shoot the ball in the basketball. Is the same DNA that he has in the muscle that gives the power. But how is it possible that DNA will turn out to be so different as a fibroblast or a neuronal retinal cell? This is based on one thing, which is the genome that is way more complicated than just 2 meters of DNA. DNA is organized in chromatin, and those 2 meters contain large amounts of proteins. And these are organized in the nucleosomes, shown here. Nucleosome is based on 8 histones, 4 dimers. And please note these tails down here sticking out from each single histone. I'll talk about them in a minute. DNA wraps around every single nucleosome twice, and this happens every 147 base pairs all along the genome. The question is, while this compaction of DNA occurs and then superstructure is organized, highly dense, the question is, how is it possible that we have physiologically meaningful and time-specific activation or silencing of gene expression? So a large amount of epigenetic control goes to chromatin. This organization is called chromatin. And we wonder about what are the mechanisms that allow chromatin to be either open or closed for gene expression. So you remember those tails I showed you. This is an example of a tail of the H3 histone, one of the histones composing the nucleosome. And this is the tail sticking out from each single group of proteins. Now, these are platforms where a number of post-translational modifications can occur, either acetylation, methylation, phosphorylation, and so on. And the presence of those specific colony modifications plus their reciprocal organization will define whether a specific site will be either open or closed for transcription. And I'm going to go through these details. It's not the point here, but what I want to tell you as a concept is the following. What are the enzymes? There are enzymes doing the job of adding an acetyl or a methyl group. These are called either writers or erasers. That's the way we like to call them. A writer will add, let's say, a phosphate group or acetyl group or methyl group as examples, and thereby these enzymes will be called acetyltransferases, methyltransferases, or kinases. The eraser will do the reverse job of removing those, and thereby called HDACs, demethylases, and so on. There's another group of proteins called readers. These generally don't have enzymatic activity, but what they do, they recognize those modifications through specific domains, bromodomains, chromodomains, and so on, and through those will connect to the rest of the nuclear machinery, let's say including the transcriptional machinery and so on. So the concept number one I want to convey to you is the following. These tails are like platforms where all these modifications can occur, and through those modifications we change a specific locus of the epigenome to be open or closed for a gene expression. And then these are the enzymes, but those enzymes, in order to do that job, to add the acetyl groups, to add the methyl group, they need metabolites. So the concept I like to convey to you is that the epigenome actually senses cellular metabolism and is able to use for its own purpose, by modifying tails, the amount of metabolites in the cell. Let's say you need a lot of acetylation. You will deplay, you use a lot of acetyl-CoA around as a major source of acetyl groups in order to obtain those acetylations. Well, where do those metabolites come from? They come from these guys, metabolic pathways, which I'm sure you all know well. The Krebs cycle, the urea cycle, glycolysis, so on. So what we did in this slide is to add a star on every single enzyme that is known to be working in a circadian manner. And as you can see here, basically every single metabolic pathway is circadian. Now, what is circadian? The word circadian comes from Latin. It means a circadian. Those are cycles of about 24 hours. And they're highly conserved all the way from cyanobacteria, plants, invertebrates, and of course vertebrates. So they're extremely conserved through evolution. And they use Zeitgebers, this is a German word, means time givers, in order to be intrainary adjusted. And the most important Zeitgeber is light. That's exactly what's happening to me, let's say, since I have 11 hours of jet lag in California. And my internal clock is adjusting to the Saudi Arabian light-dark cycle, which is kind of a difficult thing to do, but I'm trying my best. Now, from a molecular standpoint, the clock is organized in general by transcriptional translation feedback loops. Then I'll tell you in a minute. What is really interesting is that those loops are highly conserved structurally and sometimes only conceptually between species. And the genes that are involved in the clock control have been cloned. The very first one was per period in the fly by Sylvan Benzer in the 70s. And then after that, many other genes will be cloned in mammals and other species. And the key thing is that what they control a large number of clock control genes of CCGs. Where is the clock in mammals? The central clock is in the brain, and light hits the back of the retina, eating specific cells called ganglion cells. And the light signal travels the retinal hypothalamic tract all the way down to the hypothalamus and controls this group of neurons called the suprachiasmatic glucose. There are about 20,000 neurons like this in every mammal. These neurons have the amazing capacity of being oscillating with the 24-hour cycle no matter what. You can place them in culture, they will still oscillate. Now, additional experiments by a number of laboratories have shown that in reality there are clocks everywhere, even in the periphery, in the liver and the heart and so on. And what we know today is that there's like an orchestra of clocks in every body, in every organism, where the SCN in mammals is the central organizer and synchronizer for the peripheral clocks, which, however, can be uncoupled from the central clock by various ways. One is alternative rhythms in activity, or either restricted feeding, like, let's say, what I'm doing right now by having lunch at the wrong time of day for me. All right, so what is the basic of the clock and how it works? So this is now from a pure molecular standpoint. You have transcriptional activators called clock and VMAL, and they direct the expression of repressors called purr and cry. Now they will fuse now together just for simplicity. You'll see that happening in a second. There you go. So clock and VMAL will transcribe purr and cry. And this is now daytime. This is the sun. Purr and cry will encode the repressors, and these repressors will dimerize. And then migrate into the nucleus. So you have a number of possible levels of regulation. One is transcription, of course. The other one is translation, and there nuclear translocation. By the time all the repressors are in the nucleus, it's nighttime, it's the moon. So what happens is the repressors will shut off their own expression by blocking the activators. Then there's another level of control. You see that there's a specific disruption in the destruction of the repressors. And this happens to a specific program of a proteasome. There are F-box proteins specific for the clock system. By the time all the repressors are gone, it's daytime again, and this happens every 24 hours in every cell in our body. So what is happening when you destroy the system, when you destroy this beautiful molecular machinery? Lots of problems. You have increased insomnia, depression, metabolic disorders, inflammation, obesity and diabetes, even examples like accelerated aging, and of course even cancer. So you can see that disruption of the clock is a problem. Now, yes, there's a lot of information about when you disrupt the clock system by mutation in the mouse. You can see all the different type of metabolic disorders you have. So how this works and why this is important? It's important because we believe that the connection between the clock and metabolism is not only transcriptional, but also is epigenetic. And this was shown by this particular group of experiments I'll show you very quickly. So the question is, we have something like 15% of our genome that is controlled by the clock system. And this goes through, in generally thought, to go through E-boxes. This is the most common promoter element in the genome. So what really happens is the oscillation of gene expression, and thereby a beautiful harmonic cyclic expression through a mechanism of gene expression. However, as I told you, 15% of the genome is a lot, and we thought had to be connected somehow to changes in chromatin level. And this is an experiment that started several years ago by a postdoc great postdoc from Japan, Masao Doi. Masao, what he did was to take now an antibody, well, recognize the tail of H3 only when it's acetylated in K9 and K14. When he uses that antibody and recognize the specific site, you will see that on a promoter of a gene that is normally oscillating like this one, and that acetylation of that tail also is cyclic. And what he discovered that the enzyme doing that job actually is clock itself. So clock, when we thought being a simple transcription factor, is also writer, is also histone acetyltransferase responsible for the acetylation at circadian sites. Additional experiments by a number of other fellows, Junior Oyama and Yasu Nakahata, I have no time to go too many details, but found that in reality the clock system, all right, is connected to another really interesting controller of chromatin, and this is an HDAC, so an eraser called SIRT1. Now, sirtuins are really interesting HDACs because they respond to changing level of NAD in the cell. Well, we thought at the time that we have a complex that is clock and SIRT1 together, whether the balance will go towards the writer, we have acetylation, and thereby active transcription. Whether we go towards the SIRT1 level, we have silencing because the deacetylation event. Additional experiments by Yasu found also that in reality, when you look at the acetylation changes, believing that only clock and SIRT1 are implicated was a problem, because when you go and look at the levels of clock and SIRT1, they generally do not oscillate in most cells. So how is it possible we have oscillation in the readout of acetylation, while instead we have basically no changing the protein level? And that comes to the problem of metabolomics and metabolites. Well, we found that it's really NAD that is oscillating. And so it's the metabolite NAD that is oscillating that then tells the protein SIRT1 to start to be oscillating in terms of enzymatic activity. And this was what we did, and thereby we connected the enzymatic feedback loop of NAD synthesis to the transcription feedback loop of the clock system. Now, at the same time, more or less, another postdoc in the lab, Kristen Eckelmahan, asked the question, if NAD as an enzyme, as a metabolite is oscillating, how much of the metabolome is oscillating? And this connects to the talk by Dr. Fornassi this morning. So what Kristen did was to take now liver samples at different time of day and night and did a metabolome by mass spec. And we were able to read something like about 600 molecules, 600 metabolites. And you can see different groups of metabolites will have different types of peak during day and night, whether you look at amino acids, nucleotides, carbohydrates, lipids, or xenobiotic metabolites. It's a different type of peaks. All this was a huge amount of information that we were successful in putting together into some type of form. But the most important thing to say is that what we found is that of all those metabolites, about 50% of them oscillate, which is really remarkable. I mean, it's like we have half of our metabolome that is not flat, but is oscillating in a cyclic manner along the circadian cycle. And using the specific knockouts for the clock system, you can also see that a good chunk of this 50% of metabolite oscillation is clock-driven. Now, I don't have to go through all the situation here, but all this is now in a website and a resource that is called Genomics. You can go and take each single metabolite of your interest in a number of tissues and know whether it's oscillating or not oscillating, whether that oscillation is clock-driven or not. So I strongly invite you to go and do this. And you can do like NAD, uracil, whatever you like. And what we found is that we could look at the specific oscillation of metabolite and connect a specific metabolite nodes to specific transcription nodes and find that there's a coherence between those two. I don't have the time to go through all those details, but I can definitely tell you more about later on if we want to discuss. I take these last few minutes to tell you another story which is going to come out very soon, and I thought could be of interest specifically to this group of people. It has to do with nutrition, and has to do with the problems of diabetes and obesity. And this has to do with the concept of reprogramming of the circadian clock by nutritional challenge. The paper is going to come out, I think, in 1 or 2 weeks in Cell. Now, the thing is the following. How much when we eat different amount of food or different types of food or different time, this will change our clock, and is the clock able to adapt through its own intrinsic plasticity to it? Well, we've done one experiment that is designed here. What we did was to take mice in normal, chow, or high-fat diet, and this is done for about 10 weeks. And then we take the mice at the end of this 10 weeks of either normal, chow, high-fat diet, and we sacrifice them every 4 hours along the circadian cycle, covering a cycle and a half about. And then we took all samples you want. We took liver, muscle, serum, and so on. And then from those, we done the transcriptome and metabolome analysis by mass spec. Of course, I have no time to go and tell you all the data we got. But just to give you a couple hints about the results we obtained that I believe provide some really interesting insight. Well, first thing we can show you is that when you go and look now at groups of metabolites that oscillate in normal chow versus how they oscillate in a high-fat diet, you can see just visually that when you take groups of metabolites of mice being under high-fat diet, that dramatically switches the groups of metabolites that will oscillate at different time of day and night. This now doesn't tell you a single metabolite when is oscillating. This tells you how the abundance of metabolites at different time of day and night. But of course, we can tell you precisely each metabolite what it does. And again, I don't have the time to go through all the details, but you can go and look at a couple of examples as shown here that I think are really revealing. Well, I don't have to tell you all this. This is now normal chow, high-fat diet, different time. There's Zeitgebers along the circadian cycle. And you can see all different nice metabolites which will oscillate. I think the top here is sorbitol, and then stops to be oscillating in a high-fat diet. But a couple examples I think are really revealing here. One is NAD. I told you NAD is controlling sirtuins. So controlling HDAX is controlling the deacetylation of part of the genome. And yet, when you put this mice in high-fat diet, you can see that while in normal chow, you have the normal nice oscillation of NAD that we already reported. That oscillation is completely abolished when you put mice in a high-fat diet. So what you do when you have a high-fat diet, let's say you have too many burgers, too much pizza, that goes for too many days, what you're really doing, you're disrupting not only gene expression, but you're disrupting also the control of a number of enzymes like the sirtuins and NAD-depending enzymes. There's another important metabolite called uracil that we described already in the past to be oscillating very beautifully in normal chow. You can see that oscillation is strongly dampened, and this is, of course, critical for DNA synthesis in DNA metabolism. Now, the surprise came from this experiment. This is now transcriptome analysis, and this is now normal chow experiment, and this is a high-fat diet. You can see about 1,000 genes here, beautifully oscillating by groups. You can see now different groups oscillating different peaks, different Zeitgebers. The oscillations of the dose is dramatically disrupted. That was that was like, let's say, expected. We were not surprised to see this, but we were surprised to see something else. When you go and take now another group of genes, this is another 1,000 genes that will be normally not oscillating in normal chow, they start to be oscillating in a high-fat diet. So what a high-fat diet is doing is not to disrupt, it's not simply to disrupt the normal cycles of our physiology, but it is installing a new program. There's a new clock that is now starting to click and to tick in the high-fat diet liver, and this is now a new type of clock, and of course we know precisely what those genes are. And you can imagine this being part of the inflammasome at least. So I'm not going to go through too many details, but I decided to show you two examples. These are two genes of this group, SIDAC and PCX. And we wonder about how is it possible that genes that were not oscillating before now start to be oscillating? Do they have E-boxes? Do they have the normal sites where clock and BMAL will generally bind? The answer is no, they don't have clock and BMAL. They have other promoter sites, and what we found, and this is only part of the story, but at least in one case, the high-fat diet that disrupts circadian function in the clock is, okay, I'm going to skip this, is controlled by PPAR gamma. Anyway, so the picture is the following. Loss of oscillation, we have two pictures. Loss of oscillation, those are all the transcripts will start to not oscillate anymore in high-fat diet. Those ones do not oscillate anymore because clock and BMAL are not able to be recruited to a promoter anymore, and thereby the oscillation-specific metabolite becomes flat. That is the example of NAD I showed you. So normally clock and BMAL controls the nicotinamide phosphoribosyltransferase transferase, the enzyme critical for NAD synthesis. That's a normal situation. In a fat diet, clock and BMAL are not recruited to that promoter, and thereby NAD becomes flat. In the case of a gain of oscillation, now that all those genes I showed you become oscillating, at least one player is PPAR gamma, a critical element of differentiation of adipocytes, and as well as a critical element in inflammatory response. In normal chow, PPAR gamma protein is flat, and thereby some key genes controlled normally by PPAR gamma are not oscillating, and thereby also the metabolite controlled by these genes, SAHA, critical enzyme, critical metabolite from the metabolism methylation, is flat. In high-fat diet, PPAR gamma becomes oscillating, the gene becomes oscillating, and otherwise also the metabolite becomes oscillating. So PPAR gamma is recruited in a circular fashion. All right, summary, basically what I showed you is that nutrition is critical for our physiology, as we all know. I think the next speaker is going to tell you how important is the way our nutrition is changing our society. We've got to be very careful in what we eat and how much we do that, but also when we do that, because the way we get food will challenge our system, and if you have the wrong food, that might reprogram the circadian clock. It's not just a matter of disruption, but we'll reprogram it in a way that will make it different, and will make it more towards an inflammatory type of situation. The reprogramming involves a number of pathways. We unravel the Kaplan that will be published soon. As I said, these include both epigenetic and transcriptional pathways. Reprogramming is nutrition-driven, is independent of obesity. I didn't show this data to you, but also we found that the clock is very plastic, and in fact, that reprogramming is reversible. We can put these mice that are in a fat diet back in normal chow, and the previous program will be studying it. And of course, as a last point, this tells you that depending on the nutritional epigenetic context, the circadian epigenome and the matrix of the circadian clock is controlled by the circadian machinery might change and become more expanded. So just to finish, I hope I convey to you that the connection between nutrition and metabolism and epigenetics, and thereby how sometime we believe that this effect could be even transmitted through generations. We believe that a good chunk of that connection is played by the circadian clock machinery. I'd like to thank the people in the lab. Again, this is not far away from the campus, but mostly I'd like to thank Selma and Kristen right here, although everybody really contributed. Thank you for your attention.
Okay, thank you. Are there questions from the audience? Anyone? I guess one question that I think of is, this is probably really, high-fat diet perturbs the redox state, and so the question is, how much is oxidative stress, impacting this either as triggering some of these events or perhaps being downstream? I imagine with these major changes in metabolism that you're going to have major effects on redox signaling.
Of course, we do know that very well. In fact, the genes that we see activated, as well as the metabolites that we see now changing, reflect that picture. What we were not expecting was the possibility of the plasticity of the system. to be readjusting to a new type of cycle. That's to say, do we expect the obese people, people are over fed, will be disrupted in their circadian cycle? They're not disrupted. They are different. That's the concept. And I believe that's an important concept because from a pure pharmacological point of view, I will imagine different type of strategies for a different type of people.
Is it fat or sugar? Is it fat or sugar in the diet?
I cannot hear you.
I'm just wondering whether it's the sugar or the fat in the diet that actually make a difference.
That's a good question. My picture right now is sugar. The reason I'm telling you this is because we've done some experiment with ketogenic diet, which is high in fat and low in carb, and we don't have the same type of picture. So I believe different type of diet are going to induce different type of ripple effect.
I have questions. As you mentioned that there is a relation between nutrition and epigenetics. Can you comment if there is any relation between Prader-Willi syndromes and the clock center, I think?
What did you say?
Prader-Willi syndrome.
Oh, sure. No, there is one, of course. Prader-Willi syndrome is one of those diseases that has a very strong epigenetic basis. Now, whether that has a connection with the disruption of the clock, there was a couple of reports showing that in mouse models, showing that, unfortunately, those reports are only limited in quality, so that the only thing these people did was to look at the motorical activity of those mice. But yes, they did have some disruption of the clock system.
I want to know what is your opinion about paleo diet and no sugar, no grain diet?
I really cannot understand. What is my opinion of what?
About paleo diet or no sugar, no grain diet. Only fruits and vegetables.
So if it's good to eat fruit and vegetables, that's the question. Yeah, it is good. I cannot hear the question properly. But I'm not a nutritionist. I can only say that a balanced diet is the best thing you can do. And definitely, vegetables such as broccoli and so on are critical antioxidants. So definitely you can go that way.
Okay.
Sorry, one more question if I may.
Last question.
Following on from the previous question, may I please ask you what your opinion is about a diet rich in fruits and vegetables and potential effects of phytochemicals? For example, compounds such as resveratrol might have on SIRT1 activity and NAD levels in the long term, comparing a high-fat, high-sugar diet, which is processed, and a traditional, healthy, and balanced diet full of fruits and vegetables.
All right, so that's an interesting question. Two things to say. One, I believe the next speaker will be more helpful in that direction. The other is that we are thinking that in that sense, with experiments in the lab, and we found that a diet that is rich in some antioxidants. Okay. In some antioxidants will be actually positive for sirtuin activity.